Protein is essential for life. We need protein to build muscle tissue, to make enzymes for our metabolic activity, to form the protective layer of our skin, to make DNA. In fact, for most processes in our body, there are thought to be 19,000 different proteins in the human body, all with different specialized functions and locations. Most people get a lot of their protein from their diet, specifically from meat. But what about the rest? In this video, we're going to look at a mycoprotein, which is protein derived from fungi rather than animal tissue or other better known sources. So let's row back a little. What is protein? A protein is a molecule made from individual subunits called amino acids. Our body needs 20 different amino acids to make up all of the different proteins inside it. Our bodies can synthesize 11 of these amino acids itself, but we need to take 9 of them in through our diet. These 9 are called essential amino acids. So we consume proteins in our diets, which contain different combinations of amino acids. They are broken down into amino acids, and then our body can make all the proteins it needs to function. Animal products, like meat, poultry and eggs, have high levels of protein and a mixture of different amino acids, and so people who choose to eat these products are able to meet their protein intake requirements. But there are some people who do not want to consume animal products. Vegetarians, vegans, people who have an allergy, or just don't like eating meat. How do these people get enough protein and a balance of essential amino acids in their diet for their body to function properly? One answer to that question is mycoprotein. Mycoprotein is a protein that is derived from a species of fungi called Fusarium venenatum. Let's have a look at how mycoprotein is made. The fungus is cultured in industrial fermenters, which is basically a large container of fluid and microbes. The fermenter is filled with a sterilized water and glucose solution. A starter culture of the fungus, Fusarium venenatum, is added, which use the glucose for respiration. Over the next 5 weeks, more sterile glucose, ammonia gas and oxygen from the air are added to provide the fungus with the right conditions to grow. The ammonia is a gas which contains high levels of nitrogen. The nitrogen is used by the fungus to produce amino acids, which in turn makes up proteins. The oxygen in the air and the glucose allow the fungus to respire aerobically. Over the 5 weeks, given the optimum pH balance, temperature, nutrient concentration and oxygen conditions, the fungus grows rapidly, doubling its biomass every 5 hours. The fermenter has a cooling system to remove excess heat generated by the respiring fungus. At the end of the 5 weeks, it's time to harvest the mycoprotein. Before it can be used to make different kinds of food, the mycoprotein has to be heated up to 65 degrees to remove nucleic acid. The nucleic acid is removed because too much of it can cause problems for people with gout. The product is then dried to remove the water and chilled, and voila, we have mycoprotein. Once dry, the mycoprotein can be used to make vegetarian versions of meat products, like vegetarian sausages, burger patties, or mince. Mycoprotein supplies all the essential amino acids that humans need to make proteins. It is also low in fats, so it can be a healthy protein source in our diet. As well as being a good source of protein, mycoprotein is very efficient in terms of growth and has a reduced environmental impact compared to traditional meat protein sources. As mycoprotein is grown in fermenters with controlled conditions, it can be grown in a laboratory anywhere in 5 weeks. And that's it. Easy-made mycoprotein can be a valuable source of amino acids that the body needs to make all kinds of different proteins, especially when considering growing world population with a greatly increasing protein demand.
If you liked the video, give it a thumbs up and don't forget to subscribe. Comment below if you have any questions. Why not check out our FuSchool app as well? Until next time.
From part 1, we discovered how to culture bacteria ensuring there is no contamination. In this video, we're going to look at how we can calculate the size of our newly cultured populations. A bacterium has a mean division time of 20 minutes. Starting with one bacterium, how long do you think it would take to produce 1 million bacteria? Pause the video and see if you can work it out. Did you think about 6 hours 40 minutes? The working on the right requires mathematical knowledge of logs, which you don't need to worry about learning. The table on the left works well enough for us. I'm just showing you this for anyone that does know about logs so you can see them being used in real life. What about this question? Pause the video, work it out and click play when you're ready to check. Did you get 8 cells in about 5 hours? Bacteria are becoming resistant to antibiotics, and so choosing the right antibiotic for the specific bacteria is very important. A recent report suggested that by 2050, 10 million people worldwide may die every year from diseases we can no longer cure due to antibiotic resistance. This agar plate with our bacterium on has discs with different antibiotics and concentrations placed onto the bacterium. After time, the colony now looks like this. To measure the area of this clear zone, we need to find the diameter and use the area of a circle formula. So we measure the diameter of the clear zone, which is 2 centimeters. If the diameter is 2 centimeters, then the radius is 1 centimeter, because the radius is half of the diameter. So the area of our clear zone is pi times 1 squared, which is 3.14 centimeters squared. Can you work out the cross-sectional area of this colony? Pause the video, work it out, and click play when you're ready. Did you get 12.6 centimeters squared? So now we know how to calculate the number of bacteria in a population after a certain time, using the mean division time and listing out the number of bacteria and time in a table. We can also calculate the cross-sectional area of colonies or clear areas around colonies using the area of a circle. Pi times